goodness. Oh my goodness. Can you believe this? Every single piece is busted. Oh, that's got to be fixed. Holy cow. Can't believe it. What's that? Oh, the markers suck. Markers suck for me. I have to alternate markers like every few minutes because the ink doesn't replenish fast enough. Yep. Yeah, I've got my piece here from Monday night, so that'll work. That is a bummer. I mean, there's some decent sized pieces here that we can get by with. Anyway, grading the exams. I finished the practicals, so I've got all those grades recorded. I'm working on the lecture exams. Those will go back on Friday. They will go back on Friday. We'll be done with them. And I don't know if I'll have it up. The goal is when I pass them back on Friday, I'll have in the Blackboard gradebook, I'll have what I currently have you at for lab, lecture, and then the overall course grade. So we can see. Now, with those, the grades do drop I do drop the lowest quiz for both lecture and lab, but I don't think I'm gonna have these dropped. It's kind of at the end of the class. It's kind of hard to have Excel have the function, the formula in Excel actually do it before we have all of the grades in there. So this will be everything that we have with the breakdowns. We still have exam 3. We still have the final exam. Final exam will replace the lowest exam grade. We do have Tri-Beta blood drive should be coming up soon. I will offer some extra credit points for that one. I don't know the date of that, but if you try to go donate and get rejected, if you get somebody to donate in your place, that'll work. Or I'm sure they're going to need some volunteers to work the desk. If you volunteer and work the desk, because you can't donate, that will work. You also do that, and then we have another extra credit opportunity. This one is a story. Where I kind of like the diagnosis parts on the exam. It is like a one-page story of a poor sap that is hit with at least one parasite. And it's your job to kind of go through it and make suggestions on what they could have and why. Like, why do you think it's that based on the symptoms? That'll get posted in the next couple of weeks. And that is due. I think that is due when you come to the final exam. That's when you'll turn it in. And I have to double check. I want to say it's at least 5 points. I think it's at least 5 points. But it's you get the number of points based on how well you did on identifying these things. So that's coming up. That is coming up. All right. We're not going to drop the board because we're getting ready to do some life cycles, but we're on the Acanthocephalans. We went through the generalized life cycle. Remember the progression: egg, acanther, acanthocephala, cysticanth, and then the adult. We're always going to proceed through these steps. So if we only have a 2-host life cycle, which I would say is probably a little unusual, or actually it's not. If you have paratenic hosts, that will serve that kind of bridge our gap. So paratenic host is normally where you find the cysticanth, and then the intermediate host is where you find that acanthocephala. But if the acanthocephala is around long enough and can transmit, sometimes you'll find those cysticanths in there because the acanthocephala is matured as cysticanth. So moving forward, we left off with our diversity. All right, and the common theme was manipulation of host behavior. So what we're going to do is start with polymorphous paradoxus. To this. Both of these life cycles that we're going to go over are fairly simple. Fairly simple. But there's some points to discuss regarding their adaptations. Polymorphous paradoxes. It's Acanthocephala. Surface feeding ducts are the definitive host. Also called dabbling ducts. Dabblers are surface feeders. Feed near the surface. Muskrats can also serve as a host. But our dabbling ducks are going to be the definitive host. We have our adult acanth, and it's going to be in the intestine. Usually it's going to be in the small intestine, but it can get pushed based on number that are in there. All right, so we are infected in feces of our duck. We pass the egg. All right. The egg goes out into the environment and then is consumed by our intermediate host, which is an amphipod. It is a crustacean, microcrustacean, part of the zooplankton. Invert zoo covers these crustaceans. We have them here. We can go out and pick them up. So these amphipods consume, egg hatches in the intestine. The acanthor penetrates out of the gut into the hemocoel, where it develops into the acanthella and continues development into our cysticanth. All of this is in our hemocoel, our body cavity. All right, and once we get to the cysticanth stage, we are now infective. We are accidentally consumed by our surface-feeding ducts. Why accidentally consumed? They're not trying to get the infected individuals. However, this is their food supply. They are feeding on these microcrustaceans. They're also feeding on some vegetation as well. Adaptations. Adaptations for successful transmission. When we think about transmission, right, there's a lot of different adaptations that we've talked about. We've talked about increasing production, egg production, right? We've talked about timing of release and so forth. Well, here it's mostly host behavior manipulation. And that manipulation comes with the amphipod. So our amphipod is going to be positively phototactic. What does that do? Bring it up to the surface when? During daylight hours. Who's up near the surface? Surface-feeding ducts. But it's not just light. So we know we can put these infected ones in, let's say, a fish tank, and we can shine a light on one side, and they will aggregate near that light. They swim. The infected ones will swim towards that light. They are also negatively geotactic. So they're swimming against gravity, which also puts them up near the surface. So you don't need daylight hours to see them. A lot of these surface-feeding ducks are feeding, dawn and dusk, where you don't really have a lot of light source. Those two things move them up near the surface. It increases contact with the ducts. But that's not it. They cling to surface vegetation. Bit of unusual behavior for these amphipods. These amphipods would rather be swimming because if the water gets disturbed, if they sense a disturbance near the surface, their natural response is to dive deeper, try to avoid whatever's near the surface. Well, they have a tendency to cling, all right, and hide on to the vegetation. I said some of that vegetation is a food source. They're going to be more likely to get consumed if they're attached to that vegetation, whereas if they're not, and the duck brings up the vegetation, they could just kind of wash away with the water that doesn't get into the mouth. Now, I mentioned this diving. They don't exhibit that. So, uninfected ones, when the surface gets disturbed, they dive. That's an escape behavior. These guys don't. You get the splashing, they're more likely just to kind of go along, do their normal business, which makes them more likely to be infected. All of this is caused by the parasite. Questions?
What was the other term you used for surface feeding ducks?
Dabbling. You're not in ornithology.
No.
Who's in ornithology? Or took it? That was fall? I think it's fall. All right. This is all caused by the parasite. How do we know? How do we know? Well, because we don't actually know the mechanism. That's always one of these difficult things to do, is try to figure out what's causing this change in behavior. What's the actual mechanism? We can associate it with the parasite because we have infected versus uninfected, but trying to figure out what's actually happening, that's another thing. Here, we know that there is disruption in the serotonin pathway. So serotonin is a neurotransmitter. Right? It's going to cause, for us, serotonin is what our happiness transmitter, right? Is that right? Right? That's why, some people that eat chocolate and serotonin receptors, they flash off. It's like, ooh, enjoyable, right? You can't say that these guys are actually thinking, oh, great, I'm feeling great. We don't know that. But it is a neurotransmitter. So what we could do is take some of these uninfected guys, inject them with the same type of compound that gets released by the acanthocephalans and produce these same type of behaviors. And that compound, when they started doing structure analysis, said, wow, this looks like it could be as a serotonin. It would be recognized by a serotonin receptor. And yeah, sure enough, it is. So we actually know that here's this parasite. It's going to secrete this molecule that causes a change in the serotonin receptor pathways and the responses and so forth. So we know that. And that's what I have here. Serotonin-like immunoreactivity in the third thoracic ganglia of non-parasitized and parasitized. So you can see whatever's happening, the parasite is just causing these to flash. There's a lot of stuff happening here. So polymorphous paradoxus. We don't know the actual trigger for most of the things that cause a behavioral change. But this one we do. Questions? All right, so polymorphous paradoxus. That's example number one. The other example is Plagiorhynchus cylindricaeus. So this one uses isopods. So let's do this life cycle. its definitive host is a passerine bird. If you want a specific host, we can say robins. American robins were very common up where I grew up. I would almost say that they were more common than the stupid doves that are around here. That could have just be what I remember. Robins, all right? Our adult is in the intestine, typical. All right, so we're in a terrestrial type of system now. So just like the other life cycle, egg's gonna escape with the feces, gets passed. Eggs out in the environment, it's going to have to be consumed, and it's consumed by terrestrial isopods. Terrestrial isopods are what we would call our pill bugs, or I should say, our pill bugs are terrestrial isopods. That's isopods. All right. Inside, the egg hatches, same type of thing. Acanther comes out of the egg, burrows out of the gut, starts developing to the Acanthocephala, and proceeds developing to the cystocanth. And that all occurs in our hemicele. So, again, same type of pathway, same pattern, just different hosts. And then our terrestrial isopods have to be consumed by our robins. And yes, terrestrial isopods are a food source for the robins. Again, common theme across all of our Acanthocephalans is modification of host behavior. So what was the change? It occurs in our terrestrial isopods. We're trying to increase the chance that we're getting consumed. What do we know? And this was identified through a series of experiments, choice chambers and so forth. And we've tried to do some of these choice chambers in the ecology lab, but they haven't really worked out as well as we want. I think there's a lot of problems with those. What do we see? If given a choice, infected individuals prefer a light substrate as opposed to a dark substrate. So if we give them the black surface top here, and we put our white tables right next to them and let the isopods run around all they want, infected ones prefer the light ones, non-infected or uninfected prefer the dark ones. Why? Yeah, pill bugs are a dark color, right? Black, dark gray, whatever. If you put them on a white background, they're going to stick out. The birds are going to see them. Birds are going to pick them off. All right, so that's number one. Number two, infected individuals prefer lower humidity. And I'll punch in the next one. No preference for color. All right, so these two things are kind of coupled. All right, so normally these terrestrial isopods prefer moister environments, more humidity in the environment. Why? Why is that? Nobody in here has had invert zoe, have they? I think that's this fall. I think it's this fall. I think entomology was last fall. So isopods are crustaceans, right? These are terrestrial isopods. How do they breathe? Any guesses? No guesses? What if I told you they had gills? One, would you believe me? Good, good. Yes, you believe me. All right, so why would they prefer humidity? Yeah, keep from drying out. So the weak point in terms of desiccation are those gills. That's where they're getting their oxygen. All right, so they're going to prefer humid environments over less humid environments. Now, I also have this other thing, which is no preference for cover. So in that experiment, so in the humidity, you put up these two chambers. You've got a humid chamber, you have a dry chamber. More than likely, the uninfected ones go to the humidity chamber, the uninfected ones go to the drier chamber. We see that pattern. But then also, if we set up over on our habitat, if we set up, let's say, a tile of some sort that provides cover here, just enough for them to get under, all right, our uninfected ones will show a preference for being under this cover, whereas the infected ones don't show any preference at all. It's a random distribution, so just whatever the area, if this covers 50% of the plate, 50% of the time they're under it, 50% of the time they're not, whereas the uninfected ones are under that cover 75% or more. What do we know about that cover? Well, if we're under that cover, we're setting up a microhabitat. Where do these guys normally live? Where do we find, if we want to go pick up terrestrial isopods, where do we look? Under logs and under rocks and leaves. All right, so being underneath the leaves, you have cover. It's going to be slightly more humid. Outside of that area, you are exposed to our birds. 3 different behavioral changes, all aimed at increasing exposure of these infected individuals to our definitive host. What's the pathway? Why does it work? Good question. I don't think they've worked it out. Could it be part of that serotonin pathway? Possibly. Possibly. That would be rather interesting to set up some experiments. All right, questions? Those are our 2 diversity examples. Now we can present some other ones. We have some acanthocephalans in the lab that are, we don't have any of these. They're all aquatic. And I'm sure they cause some sort of behavioral change in their host. High chance that they do. Anyways. All right, so if we have no questions, let's move on to our next movement. So we finished up acanthocephalans. Now that we finished them, we're going to keep the acanthocephalans on our lab list. So the 3rd lab practical will have these acanthocephalans on them. Take a look at them, use presentations, use the diagrams that we have that were posted to find structures and so forth. So this is a larger presentation. I've got like almost 30 slides here. All right. And what we're going to do is introduce the protozoans. So what we're doing is moving from these larger parasites to the microparasites. All right, these are collectively our protozoans. We're going to introduce Protozoa, and then we're going to move to our first group, which is the flagellated protozoans. And we start with our African trypanosomes. So, Protozoa. These are unicellular eukaryotic organisms. What does that mean? What does it mean to be eukaryotic? Membrane-bound organelles. Very good. All right. Would you say that they are possibly multinucleate? Could have more than one nucleus in there. Could appear for a couple parts of their life cycle. We'll see some. All right. Our protozoans, our protozoan parasites, are both endo and ectoparasites. They can be intra or extracellular too. So if we are If we are an endoparasite, we could infect cells, or we could be free in the body, in the fluid. And it's important to talk about our metabolism for these guys. So the main type of metabolism will vary based on the species that we're looking at. Quite a few of them exhibit oxidative phosphorylation. That's what we would typically associate with metabolism. So you have oxidation and phosphorylation that are linked. That's basically our electron transport chain, right? Transferring energy or electrons down to our electron receptor, right? We're generating ATP in the process. But if we're going to do this, we need a functioning mitochondria, and we have to have oxygen present. It works for us. We've got oxygen. We depend on oxygen. It's oxidative phosphorylation. But not everywhere that these guys live will have abundant oxygen. And if you don't have abundant oxygen, then there's really no point to rely on oxidative phosphorylation. But we still need some way to generate ATP. So we can have, let's say, just glycolysis. Right? Not super efficient, but you can get the job done. Or we can use some other receptor, or electron receptor, to generate ATP, and that is our substrate level phosphorylation. So now our oxidation and phosphorylation steps, they're unlinked. Right? And instead, and that means we're not dependent on oxygen for ATP generation, and that's oxygen that's acquired from the environment. We do have less efficiency because we're not doing that electron transport chain, but what we're using is some other compound during our metabolic step to produce our ATP. So this isn't just glycolysis, it's glycolysis and something else. All right, so if we don't have oxygen, we should try to think of some other ways. And we'll see both. We'll see both oxidative and substrate level phosphorylation in these groups. And we'll leave the specifics of both of these to covered in cell bio pathways. Or is that all of biochem? No. I know we covered it in cell bio, and then biochemistry was a big part of it. Which I had it in a biology department in grad school because I didn't want to take it in the chemistry department. I'm not bragging. All right, ready? All right, the reproduction. Modes of reproduction vary in this group. All right, so we have our regular asexual reproduction. Or we could have some sexual reproduction, and we'll distinguish what sexual reproduction means for our protozoans. But for the asexual reproduction, we can have binary fission. All right, so that's just like what you would remember from bacterial replication. Bacteria all exhibit this binary fission. That's always the example that's used. But protozoans can also do that. So you have a mother cell, they divide, you produce two daughter cells. So you're basically doubling everything that you have, and then you have your cytokinesis event that splits them into two daughter cells. That's binary fission. We're going to use a term called longitudinal fission, which is binary fission. It's just on a longitudinal axis is how it gets split. And then we also have something called multiple fission. Multiple fission is a situation where we have more than two nuclear division events before the first cytokinesis event. So again, if we think about what's happening during our division, during mitosis, you have doubling of the internal contents, and then when cytokinesis happens, you have your pinching in of the membrane until they fuse and punch off, and you've got your two cells. What we're having here is instead of just a single doubling, we're having more than one replication phase inside. So then when we do have the cytokinesis event, we're pinching off to produce more than 2 daughter cells. All right, so that's what this is saying. We have our multiple division events happening inside the cell itself, multiple nuclear divisions happening within this single cell, and then as soon as we start having our cytokinesis event, we're having multiple pinching ins, and each of these nuclei moving out to those pinching in out pockets. So in our end, we've got more than 2 daughter cells happening. All right, and just verbalizing it, we kind of go through. Yeah, we're going to have it share We're going to share a common cytoplasm. We're going to have more than one set of organelles. They're going to all be connected to a cytoplasmic bridge until that final pinching event happens. There are several types of multiple fission events that exist. And their name all depends on what type of daughter cell they're producing. So I think if you remember me saying the protozoan is probably the most difficult part, it's because of these terms. They're all multiple fission, but which one appears in the life cycle depends on what stages, how they name the life cycle. So if we are producing schizons through multiple fission, it's called schizogony. So schizon, S-C-H, so for schisms, breakage. If we are producing merozoites, then that multiple fission event is merogony. If we are producing sporozoites, then we are producing it's from sporogony. And in some life cycles, we will see both merogony and sporogony. So production of merozoites happening, and then once we go through our sexual phase of the life cycle, you'll have production of sporozoites via sporogony. But multiple fission is that process of producing more than one daughter cell, or more than 2 daughter cells. So that's the asexual reproduction that we'll see here. When we have sexual reproduction, we're not necessarily talking about male and female organisms and, let's say, ovaries and testes, because we're dealing with a single-celled organism. But when we do have sexual reproduction, what we are talking about is a fusion of gametes to double our genetic material. And then we're going to have a reduction to get back to our normal state. Some cases, this would be haploids. So the normal asexual phase is all haploid, and then you'll get to a diploid stage once we fuse the gametes, and then meiosis happens and it gets back to haploid. In other cases, we're talking about the normal situation is diploid, so 2N. And then once we fuse our gametes, then we become 4N, and then you'll have your mitotic event. And which one dominates it changes. It's kind of like botany. Some of you had botany. If you go into your algae, we'll do this, but if you get into some of the bryozoans, you're dealing with alternation of generations, and in some cases the diploid stage is the dominant stage. In other cases, it's the haploid stage is the dominant phase. It just varies. In our life cycles, when we talk about sexual phase, we'll talk about fusion of gametes, and then we'll talk about our meiosis followed by replication. We'll kind of point those out. Benefit of sexual reproduction is potential for a rapid increase in genetic diversity. You're doubling genetic material. You are increasing the chances for recombination events that could possibly occur, producing more diversity, and that's a pretty good thing. Why is that? What's that? To exist longer. What are they under the assault of? What's fighting them all the time? The immune system. So for these guys that are inside, they're faced with the immune system. You're faced with antibodies. You're constantly having to change or do something else to evade that host immune system. There is an exception of the fusion of gametes, and that is conjugation. That's where you have temporary fusion of two cells. They exchange micronuclei and so forth. That's observed in ciliates. I think I mentioned ciliates, but I don't think we cover any of the ciliates in this lab. So Just adding my exception. All right, our organization of protozoa. They are typically divided into 4 simplified groups, and the simplified groups are based on their movements for the most part, how they move. This doesn't mean that we have 4 phyla. We've got quite a few more phyla, but this is how we're gonna group it. So we're gonna group it by flagellates, and that's our one group. This is where we have our movement is primarily via flagellum. All right. Now the flagella does not have to exist throughout the entire life cycle. Some of our life cycles, the flagella only exists for part of it, but they still have flagella. Second group would be our amoeba. These are those that exhibit the amoeboid movements. Our third group are the opalinids. These are commensals, primarily in the lower intestines. They'll exist in fishes and in amphibians. We're not going to discuss this group. If you were to go out and look and do necropsies of frogs and do parasite community of frogs, there's a distinct chance that you would encounter them around here. We're not going to discuss the opalinids. Then we have the alveolates. This includes all of our dinoflagellates, those that have their tests, some sort of shell of some sort. And then our 2 big phyla, Acanthocephala and the Coleoptera. So this will include our ciliates and then some other things that maybe we don't know the movement. Opalinids used to be kind of grouped into the ciliates, but I think systematics has kind of separated them out. Pulled them out and said, no, they are distinctly unique. So this is going to be our organization that we're going to break them down in. We're going to hit up the flagellates, then the amoeba, then the alveolates, or really apicomplexans. Ready? So we're going to start with the flagellates. It's a very diverse group. All of them are going to possess 1 or more flagella on the pellicle surface. We're introducing the term pellicle. Again, every field just likes to come up with their own term for stuff. The pellicle is the cell membrane and all the associated infrastructure. So all of that infrastructure associated with the cell membrane and the cell membrane itself is the pellicle, that outer surface. Our flagella is a classic 9+2 structure. I am almost 100% sure that you talked about it in one of the intro bio courses. So with this 9+2 structure, what we have is a central set. You have these microtubules. So you've got basically 2 of them in the middle, and then you have your 9 pairs that are around it. And it's those microtubules that will move, that generates the movement of the entire flagella. That 9+2 structure is what we call the axoneme, the central axoneme. That is what's running through our flagella. So we've got just kind of a diagram. That entire, this stuff, that's our axoneme. All right. And then you have the cell membrane that encompasses it. And the cell membrane of the flagella is continuous with the cell membrane of the protozoa. They're not separate, they are continuous. The axoneme itself is going to arise from another structure called the kinetosome. The kinetosome is also called the basal body. I believe when you learn this in the intro biology course, basal body was used. That was the term that you used. We're introducing the kinetosome because the kinetosome is visible in some of our parasites. We can see it. All right, so our kinetosome is located in our flagellar pocket. So you can see in this diagram, we have a slight indentation. That's our pocket. It's a complex subfollicular ultrastructure consisting of 9 sets of 3 microtubules forming a tubular cylinder plus a core containing microfilaments. Big one. If we can simplify it, the kinetosome gives rise to our flagella. To the axoneme of the flagella. That's what we should say. Kinetosome gives rise to the axoneme of the flagella. And then the rest of that is textbook definition. So here's your 9 sets of 3. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 sets of 3. You've got your microfilaments in between. These will then get rearranged into your 9+2 structure. All right. So all of our flagellates have these. Ready? These are single-celled organisms. They're going to obtain their nutrients via penocytosis and active transport of small molecular weight compounds across their cell membrane. So they're basically absorbing nutrients. Diffusion. Well, some diffusion, but active transport proteins and stuff will move stuff in. Cell membrane, it's also called the plasmalemma. Again, ancient terms that still persist today. Parasitic members of the flagellates are going to infect all phyla, and they can live in different habitats of the host. This is general to kind of explain the diversity. So a single protozoan species, I'm not saying lives in multiple habitats, like where they can live in the intestine and the blood, the muscles, intracellular, extracellular. I'm saying that you're going to have species that live in the gut. You're going to have species that live in the blood fluid, species that live intracellularly in the red blood cells, some that live intracellular in the gut epithelial cells, all of that. So parasitism is diverse in this group. Speaking of diversity, our life cycle could be direct or indirect. So it could be a one-host life cycle, or it could be multiple hosts. Our parasitic members could be intra or extracellular. We already mentioned that. But our primary mode of reproduction in our flagellates is our binary fission, which is great, because if you remember lists, then okay, I don't have to mess with trying to know, is it merogeny, is it sporogeny, is it schizogeny, is it a merozoite, and so forth. You don't have to know that. It's all binary fission. It's all binary fission. That's our primary mode of reproduction. We have a couple different flagellates that we're going to talk about. All right. And these flagellates are going to be broken apart across 3 different phyla. So we're going to talk about all 3 phyla. The Euglenozoa, the Retortomonada, and the Parabasalia. So Euglenozoa, you've looked at this in zoology. You've looked at Euglena. Right. Inside the Euglenozoa is a class Kinetoplasta. This is the trypanosomes and Leishmania. They are so named because they possess a kinetoplast, which we'll describe, which is different from a kinetosome. The Retortomonada in the class Diplomonada is Giardia. You've probably heard this one. I've seen this one. Our third phyla is the Parabasalia. It's also called the Acanthocephala, based on some classifications, because these guys have an axostyle, which is like a stout medial rod or median rod that kind of provides some support against which, like some muscle, some microtubule filaments could act. We're going to use Parabasalia. In the class Trichomonadidae is Trichomonas. We're going to look at Trichomonas. And as we move forward, a lot of these we're going to need oil immersion. We're going to need the 100x objective. On the 100x objective, it's going to be hard to see some of these things because we don't really have super great scopes, and some of our specimens are unstained. So we really need some, ways to increase the contrast. So when we get to these parasites, it's going to be good to not only look at the slides and see what we could identify easily on the actual slide itself, but also look at diagrams, textbook diagrams, and how could you take a hand drawing of that diagram and label the terms that are on our word lists. In some cases, I'll take a picture, kind of a close-in picture with my research scope, and then have multiple arrows on it, and just like we've done in the past. So what we're going to do, and I think what we'll do on Friday, is start with the class Kinetoplasta. So we're dealing with the Kinetoplasts now, Euglenozoa and so forth, but we're moving to the African trypanosomes, and then we'll move to the American trypanosomes. So what I'm going to go ahead and do is open up the Acanthocephalan quiz and have the life cycle thing. But I think I'm just going to make that do like on Monday, because we're not going to get through this on Friday. We're give you a week off of taking these things. So, any questions? Yep.
So these are like much smaller than eosinophils now?
And what?
These are like much smaller than eosinophils now?
Yeah, some of these guys will infect our red blood cells. So trying to find those guys are tough. These guys are about the size of of those cells, of the red blood cells, maybe slightly smaller. Actually, they're smaller, they're thinner, and that's part of the issue. So, yeah, they're small. All right. Let's call it quits.